really happy and honored that the organizers have invited me here with the topic that I feel really passionate about. So I've had a little bit of a technical accident with most of the slides vanishing, but I've tried to recreate the set. So let's hope it goes well. So if this were an interactive session, I would have asked you what you think about drug development as a process. But since it's not, I'll just tell you that the process of developing a new drug that effectively targets a specific weakness in a cell, involving specific preclinical development and testing followed by human trials to determine the safety and efficacy of that product is all drug development entails. So just some basic fundamentals about drug development. Innovation, serendipity, and lateral thinking are the pillars of drug development. Because drugs are an integral part of the practice of modern medicine, and new diseases and conditions are being progressively addressed by developing new drugs. I think we have to know about it, and we have to focus on that with a lot more of a research mind. So for every 5,000 to 10,000 compounds that undergo preclinical testing, only one makes it to the market. And the cost of that development is approximately 500 million in today's times. It's mind-boggling. And why should clinicians be empowered to have this knowledge about drug development? Because ultimately you are going to use it for your patients and you are going to drive the drug development itself. So it promotes innovation, assists in evaluating new products, understanding the importance of reporting adverse events, which you are the primary evaluator for that, and then suggesting new medications as well. So we need the physicians to be empowered with this knowledge for patient education and conduct of clinical trials. And researchers' mindset will always help in personal growth and development as well. So honestly speaking, ये सभी बात नहीं है. Just some basics and some jargon about drug development, just some classical terms and definitions that we need to familiarize ourselves with, so that we can build on our knowledge of drug development. We all know what a drug is, and we don't need to elaborate on that. But a drug product may be described as a finished dosage form containing a drug substance, not necessarily in association with other active or inactive ingredients. A clinical trial or study may be defined as any investigation in human subjects which is intended to discover or verify the clinical, pharmacological, and other properties, including the pharmacodynamics and pharmacokinetics of an investigational agent. Something that has just been discovered, not used or approved, with the object of ascertaining the safety and efficacy for the purpose of regulatory filing, is what clinical trial is all about. New chemical entity and new molecular entities, these are two terms which are somewhat overlapping. There is a fine nuance between the two, but they are almost used interchangeably. And that, according to the US FDA that has defined it very well, is a drug that contains no active molecule which has already been approved under any other application. An IND or investigational new drug is an application or a request for the FDA authorization to administer an investigational agent to humans. So prior to initiation of any clinical trial or even transporting the drug, the IND application has to be submitted, it has to be approved, and a number has to be generated. A new drug application. So when everything of the entire gamut of clinical trials for one particular drug or drug product is completed, and when a sponsor really believes that there is adequate evidence of the drug's safety and effectiveness to meet the regulator's demands or the regulations for marketing approval, the sponsor submits a new drug application to the FDA or the regulatory agency concerned. And it must contain all the data only after the NDA is approved, you can market the drug. If you look at the historical perspective, we've come a long way in the last 100 years or 200 years. At the turn of the 19th century, we really did not have any regulations on the marketed foods or products, and there was quite a bit of exploitation and fraud. Just about anything was legal. Eventually, because of a few tragedies and loss of lives, in 1938 there was the Federal Food, Drug and Cosmetic Act, which was enacted, which is considered the birth of the modern drug development and the modern FDA regulation. So it required the manufacturer for the first time to prove the safety of a particular product that they were marketing, and it also entailed penalties for misleading labels. I think it was pretty progressive for those times. Then every 10 years there was some improvement on the drug, wherein the drugs were either classified as prescription or non-prescription or harmful, not harmful. And eventually in the 1970s, there were firm regulations that came about for adopting good laboratory practices, good clinical practices, and good documentation practices. 1987 marked the FDA being allowed to even judge the safety of the product and the effectiveness based on surrogate endpoints. Now, if you just stop back and think about one of the drugs that is widely prescribed by all in this room, metformin, and I call it the wonder drug in diabetes. You all know why. So just a moment on the history of this particular wonder drug. A compound was serendipitously discovered from the plant Carica officinalis in Europe and was used to treat symptoms of diabetes at that time and also to increase milk production in cattle. The specific anti-hypoglycemic properties were being used after the 20th century onwards, and its active molecule was also isolated by French pharmacists. So initially it was used with success, but eventually there was a lot of toxicity associated with it and it was abandoned. 1920s saw the production of bihormones, and again they were produced, used and banned because of the associated toxicity that we all are aware of. But metformin as such was produced for the first time in 1922. However, because of the insulin discovery around the same time, metformin was long forgotten and was started to be used only in the 1950s because it showed the clinical properties of bihormones that were earlier used but did not have the toxicity that the earlier compounds had shown. No elaborate trials of metformin were conducted prior to its approval and the widespread use. Even the mechanism of action of metformin was not known at that time. The effect was known, yes, but the mechanism of action was not isolated as such. It has come to light only in the recent, 4 or 5 decades. From that day to 2008, when the FDA evolved its regulations further and came out with the guidance of industry, particularly in diabetes, where drug developers were required to evaluate the cardiovascular risk in new antidiabetic agents used to treat diabetes. So we really have come a long way. So thus drug discovery and drug development has several stages, almost 5 stages. The first one we can call it the discovery or the development stage, the incipient stage, where new drugs are discovered by various means. Not just isolation from plant sources or animal sources or even marine sources, but even computer modeling is used very rapidly now to discover new chemicals and new enzymes used specifically for conditions. So new insights into the biology of the diseases has allowed this kind of progress in drug development. Many tests of molecular compounds to find potential benefits are even possible at an earlier stage before you actually test them in preclinical species or in humans. Existing treatments with unanticipated effects. This is where serendipity or close observation or monitoring comes into play. And new technologies, including new ways to target medical products to specific sites. These are highly targeted products that are being developed. So after thousands of compounds, which may be potential candidates for tomorrow's drug, are evaluated and tested, only a few make it to the next stage and are used in development. So after a promising compound has been isolated, a wide array of experiments are conducted to gather more information about it. How is it absorbed, distributed, metabolized, excreted, its potential benefits, side effects that may be anticipated, the dosage, the form of administration, how it interacts with other drugs. All these questions come to mind and they are addressed one after the other by ways of several cellular systems and even computer modeling. So the first and most important step is to find a promising agent based on the advances that have been made in various fields. Understanding the pathological stages or pathways related to the disease is equally important so that new targets can be discovered. Thus, several compounds may be synthesized and tested before a promising agent emerges. The second step will be the preclinical research. Once the compound is isolated, it undergoes at least two kinds of preclinical testing, in vitro and in vivo. Prior to use in humans, it is absolutely imperative to understand the potential of the drug to cause serious harm or toxicity. And our FDA, or the highest regulatory authority in the world, in the sense of regulatory authority with the highest standards, mandates the use of GLP or the good laboratory practices for preclinical laboratory studies. So if you have a package and it does not contain the GLP labeled studies, your application is not going to be edited by the FDA. Thus, preclinical studies may not be large, but they provide adequate details of dosing and toxicity levels. After preclinical testing, the findings or the results are carefully reviewed, and then the decision is made by a team at the drug development center whether it can be tested further in humans or not. The regulations set up these are some of the basic requirements for further study. Is the study conduct has to be good. The personnel involved in the study conduct have to be written. Facilities where the studies are conducted, equipment, protocols, operating systems, study reports, every single thing has to be documented. This is only for the animal or the rodent studies and non-rodent studies. We haven't even reached the humans yet. Thus, certain animal tests like the rodent tests are mandated prior to exposure of humans to these new investigational agents. The main objectives of in vivo testing is to demonstrate the safety of the proposed medication. For example, compounds that show positivity in the chromosomal testing will never be taken forward in today's tests because we have far more advanced drugs which are non-genotoxic. Thus, the IND application of the pre-human clinical testing application should include the chemical and the manufacturing data, animal tests, including all the results, the rationale for testing a new compound in humans, and the rationale for using a particular dose. Because it is very important to demonstrate to the FDA that you are going at a dose that is much, much lower than the highest dose tested in the preclinical studies. If the FDA is satisfied with the documentation, the stage is all set for the phase 1 clinical trials. So step 3 in drug development can be considered as the clinical research step, the most expensive, the most arduous, and the most rewarding stage of drug development. While preclinical research addresses basic drug safety concerns, concerns, it cannot substitute for human testing or evaluation. Thus, clinical research is all about the studies conducted in people. Drug developers design clinical studies based on considerations of what they want to answer with that particular trial and accomplish for the drug in each of the different stages of clinical research, and then an IND is filed. Designing clinical trials, that's a very critical aspect of drug development, and physicians play a very important role at this stage. So they are designed to answer particular research questions related to that medicinal product, and they follow a specific plan or a protocol developed by the drug developer. Prior to clinical trials initiation, researchers review previously generated information about the drug to develop research questions and objectives, deciding the following: who qualifies to participate in the clinical trial, how many people will participate, how long the study will last, whether there will be a control group or not, how the drug will be administered, and at what dosage. These are very critical questions. They sound extremely easy, but it takes weeks to months sometimes to actually address each of these. And then what assessments will be conducted, what kind of data will be generated, and how will you analyze that data. So all this is actually the pre-planning for a clinical study. And typically they evolve from early small-scale phase 1 studies to the larger, longer duration phase 3 studies. So this again is just a cartoon depicting the flow of drug development or the phases of clinical research, which I will walk you through in the next 10 minutes. So the phase 1 clinical study. So don't get confused between the stages of drug development and the phases of clinical research. We are on step 3 clinical research and phase 1 of clinical research. So approximately 2,200 healthy volunteers are adequate to establish the safety of the drug. That's the only objective of a phase 1 study. But as a drug developer and a physician, and with the curiosity building up, and with newer advances, we have so many biomarkers and so many assessments to actually evaluate the early pharmacodynamic action of the drug. Because everybody is curious whether it's going to really work in humans or not at this stage. But from the regulator's perspective, it is only safety that is required to be established at this point. The purpose is safety, as I mentioned, dose and selection, because you have to make the right doses for obvious reasons. Proof of concept, I would call that 1B study. Pharmacokinetics is extremely important, and drug-drug interactions and food effect if you are really sure, or if you have been able to show some kind of efficacy even in the first evaluation of the drug in humans. So during phase 1 studies, normally healthy volunteers or normal volunteers are recruited, but for conditions that are life-threatening, cancer, or many others, you obviously recruit the patients. There is no normal volunteer exposure to such drugs. You start with low doses and incrementally move up to doses or the highest dose level that you can use in clinical studies or in humans, given the safety established in preclinical studies. Questions regarding how it works in the body, the side effects associated, increasing drug dosage, and early information on effectiveness are all determined in phase 1 clinical trials. Also possibly helps to understand how to administer the drug, minimizing the side effects and maximizing the effectiveness of possible benefits. Only 70% of the drugs move to the next phase of development, which is the phase 2 clinical studies. So phase 1 is almost by protocol. You determine a single dose study and the single dose stage, move on to multiple dosing, establish a safety there. You could also do early, clean patient studies to establish a safety in those patients for about 10 to 15 days, and then move on quickly to phase 2 studies. Up to a few hundred people with the disease or the condition that is intended to be treated will be typically recruited in phase 2 studies. Again, it lasts for a few months to several years, but we cannot forget that the support for phase 2 clinical studies comes from the preclinical testing that has been done. So if in most countries, if you want to test the drug for 6 months in humans, then you need to generate 6 months of preclinical or rodent and non-rodent data with the same drug. The purpose of phase 2 is now efficacy and the right dosage to be taken forward to phase 3. Of course, safety is imperative, but the primary objective is efficacy. So, few hundred patients with a disease or condition intended for treatment will be enrolled in these studies and to show you whether the drug is going to be beneficial or not, with additional safety data. The results of phase 2 trials are taken very seriously because for most companies, it's going to decide the fate of their own future. Most of the go-no-go decisions for a particular drug are definitely taken by the teams at this stage, and you have the results of phase 2 in hand. Approximately 33% or one-third of the drugs that have come or discovered in the preclinical testing will move to the next phase, which is the definitive phase 3 or the definitive pivotal clinical studies. Now the stage is set. You have established the safety of your drug. You have a very good indication of the efficacy of the medicine at this point. You also know you have a handle on the doses for the clinical trials, the doses that you want to take forward even for marketing. Approximately 300 to 3,000 volunteers who have the disease or condition will be recruited. These studies typically range from 1 to 4 years depending on the preclinical and the phase 2 duration. And the purpose is now efficacy, monitoring adverse reactions, and determining the population pharmacokinetics of the drug. These studies are typically designed to demonstrate whether or not a product will offer benefit to the diseased population in general. Rare side effects which have gone unnoticed so far may be elicited also in phase 3 studies, as these are longer and larger in nature. Again, approximately 25 to 30% of drugs move to the next phase of clinical research. So, just to complete, step 4. Step 4 or the FDA review that happens after you have the results of the pivotal studies, and that's a process which is undertaken, say, at the US FDA by an entire team of different experts looking at all the possible aspects of the data that has been generated and submitted to them.